Welcome again to this meeting. So I'm very happy to introduce Serena Viti. Serena got her PhD at the University College London, working on infrared spectra of stars. And after this, she has been traveling a bit around the world. She's been a postdoctoral fellow at UCL, followed by a position at the CNR, so it's a research council in Italy, in Rome, and then returned to UCL in 2003. There, she was promoted professor of astrophysics in 2012, became head of astrophysics group in 2016. And in 2019, she was awarded an ERC advanced grant. Since 2020, she's a member of our faculty. She moved to Leiden University. And of course, at the top of her research, she's also active in education and outreach. But today's talk will be on her research and on how artificial intelligence helps us find amino acids in space. Floor is yours.
Thank you, Francesca. I will share the screen now. Okay, I hope you can all see it. Thank you very much for inviting me to give this nice short talk here at faculty level. It's really interesting to be able to present some of the research to people that I usually don't talk to. So as Francesca already introduced me, I won't really say much about myself, apart from the fact that, of course, I am an astronomer. So although the research I will be presenting will be quite interdisciplinary, my research interests are in studying molecules in space, wherever we find them, from dense gas in the interstellar medium in galaxies to star-forming regions to protoplanetary disks where planets may be forming. I'm very happy to be here in Leiden. As Francesca said, I moved here about a year ago. And I have, however, still a group based in London. So my group is still split between London and Leiden. And today the work that I will present is being in fact led by a postdoc, John Holship, which is in Leiden, and a PhD student, Johannes Eyl, who's still in London. So it's not just interdisciplinary, but it's also in the inter-countries, if not intercontinental. Okay, so what research would I like to present today? So as I said, I work in the field of what is called astrochemistry. So that's the study of molecules in space. But today I'm interested in very briefly show you a very interesting project that actually marry different disciplines, chemistry and astronomy, of course, but also data science or computer science. And a lot of what I'm going to show you today is only being made possible by the application of machine learning and statistical techniques to the study of astrochemistry. So what we're doing is addressing what I think is one of the most fundamental problems in astrochemistry, possibly in astrobiology. And the problem is, can simple amino acids, so the building blocks of life, such as glycine, form and survive, most importantly, in space? So on the right-hand side here is just really a little picture of what I call the multi-million year cycle of molecular gas. So molecules are everywhere in space, from clouds, which are fairly quiescent and dark, to regions where stars are formed and the jets and powerful outflows have developed. And during this cycle, of course, at some point, planets form. So what we are interested in understanding is whether simple amino acids can not only form in space, but can survive and possibly be delivered on Earth. Although this last part, I will leave it for you to speculate upon if you're interested in this type of research. So as I said, we are an interdisciplinary group in the sense that we involve astronomers, not just astrochemists. We have even cosmologists helping us out with the more data science aspects, and chemists and indeed data science themselves. And the future of this project, just to give you again in one slide what what we're trying to do here. The future that I foresee is the development of a novel methodology that might allow us in the future to discover new molecules in space. So as I mentioned already, what we're trying to do here is trying to understand if amino acids can form in space. But backing up a little bit, one may ask, well, why do you think that amino acids can form in space? And the answer is that, in fact, they have been found, certainly in a lot of amounts, in meteorites. So on the right-hand side here, I used the histograms from this paper, Cobb and Podritz in 2014, and I hope you can see that there is a variety of amino acids found in space. And the two that, sorry, not in space, on meteorites, and the two that we are interested in seeing if we can find in space are two of the simplest one, glycine and alanine. So I'm certainly no chemist and definitely not a biologist, but I think I'm convinced that amino acids are indeed important to organic compounds and that they are, of course, important for formation of proteins. So making this connection between the building blocks of life and the interstellar medium, it's very exciting for us. These two molecules, glycine and alanine, with the formula there that I wrote in my slides, they are, of course, the two simplest ones, but from our point of view, so from the point of view of astrochemists or astronomers, these are very, very complex molecules. And as you'll see in a minute, it's not so obvious how they can even form in the conditions of space, let alone survive. So in order to find amino acids in the interstellar medium, we need to determine what are the optimal conditions for the amino acids to actually form in the first place. We are very lucky that, in fact, not only amino acids have been detected in meteorites, but also in comets. And this is important because in this particular case, which is depicted by this nice poster prepared by Isa, in this particular case, you are actually seeing amino acids in situ, so in the comets itself. And so this gives us more strength, this gives more strength to the hypothesis that amino acids can, in fact, form and remain in space. So these are the good news. The bad news is that despite great efforts from us astronomers, we have yet not managed to firmly detect glycine actually in space, in the interstellar medium, in regions where stars and planets may form. So really, we need to go back to the blackboard or to the whiteboard and actually first determine all the formation and destruction routes for these two molecules that can actually happen in the harsh condition of space. So I keep saying harsh conditions. What do I mean by that? So let's take a step back. How do molecules in general form in space? In fact, it's not that easy, because on the left-hand side here, as a background to the nice free molecules there, you have essentially what we call a dark molecular cloud, which is the cloud where stars and planets may be forming. And in these regions, the temperatures are really low. We're talking about 10 Kelvin. The densities are not that high, again, in volumes that are about 10 to the 4 per centimeter volume. So molecules takes millions of years to actually form. And yet we do know, we have now plenty of evidence, both observational and experimental evidence, that simple molecules do indeed form in abundance in regions such as this. So carbon monoxide, for example, molecular hydrogen, in some cases, if the temperature is a little bit warmer, water, and so on. But in fact, more complex molecules tend to not form in the gas, but tend to form on the dust. So we have a little bit of dust in the interstellar medium, not much, but it's crucial. And a nice simplified picture of a dust grain, you can see it on the right-hand side here. So what happens is that the dust act like a catalyst, although I've been told by chemists that catalyst is not the right word here, but nevertheless, it's a place where atoms tend to fall on when the temperature is low, and they tend to stick for long enough to hop or tunnel, get together and form molecules, in particular, more complex molecules. So we believe that if glycine is there, is as formed on the dust, and then it will have evaporated back in the gas phase if, for example, something like the formation of a star heats the dust up enough for whatever has been formed on top to evaporate back into the gas phase. So that's how we think glycine and other complex molecules may form in space. So why can't we just, for example, study this on Earth by reproducing the conditions of space? Well, we can do that. And in fact, laboratory experiments are very successful in understanding how molecules can form on the grains. And one example, one of the most successful examples, is indeed the laboratory of astrochemistry that we have at Leiden Observatory. However, as I hope is clear from the little drawing there of different reaction paths for the few molecules depicted on the right-hand side there, surface reactions in general, and surface reactions for sure, are complex, and they can only be experimented under a certain constrained range of laboratory conditions. So in other words, if you wanted to simulate every single possible condition in space, you would need many years in a lab. So prioritization here is a must. We need to be able to tell or to inform the chemist what to look for, which reactions to prioritize. And this is what our project really is about. We have started by first applying Bayesian methods to the chemistry itself, where we essentially study the rates of the reactions on the dust in order to infer the likelihood of key reactions in this particular case would lead to glycine. And then the idea is to provide a list of reactions which are particularly important to form and destroy glycine. And of course, these reactions then would need to be studied in the lab. So what is our recipe then for determining the likelihood of molecule? Well, it has three main steps. One is to choose a relatively small and closed chemical network that you can play with. An example is on the right-hand side. For small and closed, we mean you pick maybe a molecule that has a finite number of reactions that form and destroy it. And closed in the sense that, of course, every atom that participates to this network will also be important to form other species, but there must be a sort of degree of closeness that can determine if a network is nicely self-consistent. You then, step two, obtain some priors from the observations. In other words, we do have some information about molecules observed on top of the dust grains, what we call ices. So the ice on top of a dust grain, because remember, we are at very low temperatures here. So we get some information from the observations that constitutes our observational constraints. And then we run a Bayesian inference to determine the rates of the reactions in these chemical networks. This work has been going on now for a few years and has been led, as I said already, mainly by John Horship on the left photo there, who's a postdoc at Leiden, and by Johannes Hale on the right there with the dog, who's a PhD student in data science, in fact, not in astronomy at UCL. So the products of this recipe are the following. Essentially, we study the results through posterior probability distribution, which are the three graphs on the left. Here is showing you the posterior probability as a function of a large range of potential rate reactions of, first of all, the parent species for glycine, NCH4, on the top two plots, and then the posterior probability of a particular reaction that via NCH4 can produce glycine. Depicted in a nice flow diagram, you have the more complete network that we use for glycine, and that's on the right-hand side, and the different colors of the boxes there determine, are indicating whether we have some observational constraint for the molecules or not. And what we achieved with this study is we managed to reduce the number of important reactions from 49 to 14, so we reduced the dimensionality of the problem. We found that we could break the network in smaller ones, and I think, you know, the potential of the goodness of this, I think it's obvious. If you can break them, then you can study them separately and independently. And actually, as a byproduct, we ended up determining better some fundamental chemical parameters like binding energies of molecules, for example, through this Bayesian route. Again, this, determining binding energy is a problem that is tackled by experimentalists in the labs, but it's a time-consuming effort. So, to conclude, what's the next steps? Well, the next step is more amino acids, in the sense that so far we only looked at glycine, and only under certain conditions, and more AI, in the sense that so far we use Bayesian inference, but in fact the idea is that encouraged by the fact that we were able to estimate reaction rate parameters by reducing the dimensionality, we feel that this could be even more important by basically including more and more amino acids in our networks. So sure, the plan is to include a more complex reaction network, but we believe that this will be quite slow with the methodology that we've got at the moment. So the idea is that in the near future, we will be using statistical likelihood emulation to infer rates. And in particular, to start with, we are using what is called BAMBI, which is blind accelerated multimodal Bayesian inference, which in fact combines the benefit of the Bayesian inference with artificial neural networks. So I hope in this 15-minute talk, which I believe I've been on time, I have shown you an interesting project, very interdisciplinary, which merge or marry people from different disciplines, in particular astronomy, chemistry, and data science. Thank you very much.